In this video, we are going to see the anatomy of the stomach in detail, and then we are going to see what are the secretions of the stomach and how they are regulated and how the motility of the stomach it is regulated. Stomach, it is a temporary storage tank, and the chemical breakdown of the protein, it starts here in the stomach. And stomach, it converts the food to a paste-like substance which is known as chyme. And when the stomach is empty, its volume is 50 milliliters, but it can expand up to 4 liters, and it can expand so much that it can go up to the pelvis. And when the stomach it is empty, it recoils, it comes back, and the stomach mucosa, it forms many folds. Those folds are known as ragi. Stomach has four parts. The cardial part, it is the uppermost part, and this is where the cardial orifice is. The cardial orifice is where the esophagus it is entering the stomach, and this portion is known as cardia. And then there is fundus. Behind, after cardia, there is a fundus. Fundus is a dome-shaped region, and it is present beneath the diaphragm, and it bulges superlateral to the cardia. Then there is a body. After the body, there is a funnel-shaped pyloric part, and that pyloric part, it has a pyloric antrum, a pyloric canal, and a pylorus. And in the pylorus, we have a pyloric valve, which is also known as sphincter, and pyloric sphincter. And this sphincter, it controls the stomach emptying into the duodenum. Now let's see these parts. This is the cardia. This is where the esophagus is coming. This is the fundus, the dome shaped. It is superlateral to the cardia. And then this is the body. The body, it continues into the pyloric funnel shaped pyloric region. This is known as pyloric antrum. Then this is the pyloric canal, and this is the pylorus. And at the end of the pylorus, there is pyloric valve or sphincter. And this is the duodenum. And this sphincter, when it is like the thickened layer of the muscularis externa, when it contracts, it can regulate the movement of contents when they go to the duodenum. It can, if it is open, the contents can pass, but when it constricts, the contents, they cannot pass to the duodenum. Now let's see here. This is this is stomach, right? This is the cardia. This is the fundus, and then this is the body, and this is the pylorus. This is the pyloric sphincter. This is the pylorus. And now stomach, it has two curvatures. This is the lesser curvature, and this is the greater curvature. Okay, from the lesser, and this is over here, you can see the liver. On the right side is the liver, right? It is present close to the lesser curvature of the stomach, as you can see, because stomach is on the left. And this is the spleen. So from the lesser curvature of the stomach and the liver, there is a mesentery. That mesentery is known as lesser omentum. Remember, mesentery, it is a peritoneum, the two layers of the peritoneum stacked together. And in the mesentery, the arteries and the nerves and the lymphatics, they travel to the organs. And mesentery, they serve to attach the abdominal organs to the abdominal wall, and it stabilizes them. So this is there is a lesser omentum, there is a mesentery between this lesser curvature and the liver, and then there is a mesentery which is hanging from this greater curvature of the stomach, that is known as greater omentum, and it continues with the mesentery of the intestines. All right, so this is we already discussed. It has a greater curvature, which is convex lateral surface of the stomach. Lesser curvature is concave medial surface of the stomach. And then the mesenteries. Mesenteries extend from the curvatures anterior to the stomach to other digestive organs. So there is lesser omentum, which runs from the lesser curvature to the liver. Greater omentum, it drapes inferiorly from the greater curvature over the intestines, spleen, and transverse colon. It blends with the mesocolon, which is mesentery, that anchors the large intestine to the abdominal wall. And it contains fat deposits and lymph nodes. So mesenteries, they contain fat deposits. Now let's move on to the next slide. Now the stomach, it is controlled by the autonomic nervous system. It supplies the stomach. The sympathetic fibers from the thoracic splanchnic nerves, and they relay in the celiac plexus. And the parasympathetic fibers are supplied by the vagus nerve. And the blood supply is the celiac trunk, which is branch of aorta, and then the veins of the venous and also through the veins of the hepatic portal system. Now let's see the microscopic anatomy of the stomach. So just like we said in our video in which we did the anatomy of the GIT, that it has four layers. Similarly, the stomach, it has four layers. And what are the four layers? They are the mucosa, the submucosa, the muscularis externa, and the serosa. So stomach also, it contains the four tunics or the four layers, but its muscularis externa and the mucosa layer, they are modified. Now let's see how muscularis externa is modified. Muscularis externa has circular and longitudinal smooth muscle layers, that is throughout the alimentary canal, but in the stomach there is also a third oblique layer. Okay, so the muscularis externa in the stomach has three layers: circular, longitudinal, and oblique. And this helps in the movement, in the extra movement, which help in the breakdown and mixing of the food before it goes into the small intestine. Now see how the mucosa is modified. So mucosa, it consists of simple columnar epithelium and entirely composed of the mucous cells. Okay, so there are the simple columnar epithelium is entirely composed of the mucous cells, and they secrete two-layer coat of alkaline mucus. The surface layer, it traps the bicarbonate-rich fluid layer which is beneath it. Okay, and then and why do we need so much mucus layering in the stomach? So that the stomach does not digest itself. And then it is dotted with gastric pits. In the mucosa, we will see gastric pits. There will be pits going deep in, which lead into the gastric glands. Gastric glands, they produce gastric juice. Now let's look in this in this diagram. They have enlarged a section uh, from the stomach wall over here. Now let's look at it. All right, so there is surface. We said that the stomach, it has all the layers as in rest of the alimentary tract, but two of its layers are specialized. The mucosa, so this is the mucosa. The surface epithelium, it is uh, mostly mucus glands, right? And then there are pits. Gastric pits are visible. So these are the pits going in, right? And then And you can see in this pit there are many glands. We will go over those glands in a while. And then its muscularis externa. It has three layers. It has an oblique layer, inner circular, outer longitudinal. But inner to the circular layer there is an oblique layer. Right. So these are the special features of the stomach. Now let's continue. So what are the types of the glands? So these glands, they are present in the pit, which we just saw, which was going inside. Glands in the fundus and body produce most gastric juices. So the fundus and the body. And glands include the secretory cells, mucous neck cells, parietal cells, chief cells, and the enteroendocrine cells. So these are four types of cells which we are going to see in that pit. Let's see the cells of the stomach in this. So these are the gastric pits. So in the gastric pit, this is surface epithelium mucus cell, and then these are the mucous neck cells. They secrete mucus. And then these are the parietal cells. Parietal cells, they secrete hydrochloric acid and intrinsic factor. Remember, intrinsic factor, it is the substance which attaches to the vitamin B12, and to be absorbed from the small intestine, vitamin B12, it needs to be attached to the intrinsic factor. The both complex is absorbed by the small intestine. Alone, vitamin B12 cannot be absorbed. And then we have these chief cells. They have pepsinogen, which is activated to pepsin, and it acts on the protein. Then these are the enteroendocrine cells, which they have made kind of bluish. They secrete histamine, serotonin, somatostatin, and gastrin. Now let's go over these. So the mucous neck cells, they secrete thin acidic mucus of unknown function. Then we have parietal cells. Parietal cells, they secrete hydrochloric acid, and the pH in the stomach is very, very low because of this hydrochloric acid. It is 1.5 to 3.5. This hydrochloric acid, it denatures the proteins and it activates the pepsin. It activates pepsinogen into pepsin, and it breaks down the plant cell walls and kills many bacteria, right? And then the parietal cells, they also secrete the intrinsic factor. Intrinsic factor is a glycoprotein required for absorption of B12 in the small intestines. Then we have chief cells. Chief cells, they secrete pepsinogen, which is inactive enzyme that is activated to pepsin by the hydrochloric acid and by pepsin itself in a positive feedback mechanism. Then also the chief cells, they secrete some lipases. Then we have enteroendocrine cells. They secrete chemical messengers into the lamina propria. They act as paracrines, serotonin and the histamine, and then also hormones, somatostatin and the gastrin. Somatostatin, it acts as a paracrine and a hormone. How does it act as a hormone? So it affects the motility of the all of the parts of the GIT, not only the stomach. This is a bigger view, so our mucus cells are not present here. These uh, these are the parietal cells. These purple one, they have pepsinogen, and hydrochloric acid. It is secreted by the parietal cells. This hydrochloric acid, it acts on the pepsinogen and converts it into pepsin. And then these cells, we know they also secrete intrinsic factor. And so these are the chief cells, these are the parietal cells. Then we have endocrine cells which secrete hormones and the paracrines. So one more thing about the microscopic anatomy of the stomach, it has a mucosal barrier because the stomach, it has harsh digestive conditions, and stomach, it requires to be protected. So there is a mucosal barrier which protects the stomach, and it is created by three factors: thick layer of bicarbonate rich mucus, tight junctions between the epithelial cells. They prevent the juice from seeping into the underlying tissue. The damaged epithelial cells, they are quickly replaced by division of the stem cells. Surface cells in the stomach, they are replaced every 3 to 6 days. So this mucosal barrier, this is the protective mechanism of the stomach, so it does not eat itself. Now, what can go wrong? There is a condition known as gastritis. Gastritis is the inflammation caused by anything that breaches the stomach's mucosal barrier. So, if the mucosal barrier, it is breached, then the condition is known as gastritis. Then there is peptic or gastric ulcer, and it is when there are erosions in the stomach wall. If erosions perforate the wall, they can lead to peritonitis and hemorrhage. What is peritonitis? So just imagine there is a hole in the wall of the stomach, and on the content, it is going to into the abdominal cavity, and over there it is going to irritate the peritoneum, and also the peritoneum, it will try to localize that problem, and this causes inflammation of the peritoneum. Peritoneum is the layer which is lining the abdominal cavity. It has a parietal and a visceral layer, just to remind you. This is a very painful condition, and it can get very dangerous if it spreads. And then most ulcers, they are caused by a bacterium, Helicobacter pylori. So this can be tested. A test is done, and those people who have this bacteria inside their stomach, they are more prone to develop ulcers. So and it can also be caused by NSAID drugs such as aspirin. Some people, they just take whenever there is headache or some minor pain, they just take aspirin too much. That aspirin, it can cause a peptic ulcer. This is a gastric ulcer. This defect in the wall of the stomach. There is this, you can see there is a gap. And this is the bacteria. It is present in the mucosa layer of the stomach. This is Helicobacter pylori. So this is also one of the causative agents of the peptic ulcer. Now let's see the digestive process in the stomach. Processes carried out by the stomach are that it carries out the breakdown of the food by its different types of movement, the churning and all the peristalsis movement. They are present in the stomach and they're present in more complicated form. And it also serves as the holding area for the food, and it delivers chyme to the small intestine. It denatures proteins by the hydrochloric acid and pepsin. It carries out the enzymatic digestion of the proteins. And there is a milk protein which is known as casein. It is broken down by renin in the infants, and it results in a curdy substance. Maybe sometimes you have experienced a young baby throwing out curdy kind of vomit. So that is the milk protein casein, which is turned into that curdy thing in the stomach. Now, the lipid soluble alcohol and aspirin, they are absorbed in the stomach. Stomach is not a very, I mean, it is not where the nutrients are absorbed. It is where the protein digestion it starts, and it also it churns and it mechanically it breaks down the food. It is not where the nutrients are absorbed, but lipid soluble alcohol and aspirin, they are absorbed into the blood in the stomach. And the stomach function which is essential to life, it is the secretion of intrinsic factor, because vitamin B12 it is needed for the DNA synthesis and cell division. If vitamin B12 is, if there is no intrinsic factor, the body cannot absorb vitamin B12, and that can lead to pernicious anemia. It is treated by B12 injections. So intrinsic secretion of intrinsic factor by the stomach, it is a very important function of the stomach. Now, how is the gastric secretion regulated? So gastric mucosa, it secretes more than 3 liters of gastric juice per day, and this secretion it is regulated by the neural mechanisms and the hormonal mechanisms. Neural mechanisms, they consist of both long and short reflexes. Remember when we were doing the general alimentary canal, we said there are short reflexes which are enteric because there is a gut brain, right? The gut has its own brain. So there are short reflexes, and then there are long reflexes. Long reflexes are through the vagus nerve. And vagus nerve stimulation is parasympathetic. It increases the secretion, and the sympathetic stimulation, it decreases the secretion. So neural mechanism consists of both long and short. Short one are through the gut brain or the enteric nervous system, which is present in the submucosa and in the myenteric plexus, submucosal plexus and the myenteric plexus, and the long is through the vagus nerve. Then there is hormonal mechanisms. Gastrin, it stimulates the HCl gastrin. It is secreted by the enteroenteric cells in the pet we just saw. So that gastrin, it stimulates the HCl secretion by the stomach, and then gastrin antagonized hormones by small intestine. They are also stimulated by the gastrin. So it is doing both the things. Gastric secretions, they're broken down into 3 phases. There is a cephalic phase, gastric phase, and intestinal phase. Now let's go over these phases. Cephalic phase, it is the conditioned reflex triggered by the aroma, taste, sight, thought, and it occurs before the food enters the stomach. Just like when we smell something very nice, something is cooking, it is the cephalic phase, and it triggers act via vagus nerve. Then comes the gastric phase. Gastric phase, it lasts 3 to 4 hours. Cephalic phase is very short, and it is just when the stomach is anticipating the coming of the food, because the smell of the food is coming. And the gastric phase, it is longer. It is 3 to 4 hours, and it provides 2/3 of the gastric juices released. And the stimulation of the gastric phase is by three stimuli. Distension, it activates the stretch receptors. It initiates both long and short reflexes. So distension of the stomach, And in the long reflexes, the impulses travel to the medulla and then back via vagal fibers. And then the second stimulus is the chemical stimulus. And what is that? The partially digested proteins, caffeine, low acidity, they all activate the endocrine G cell to secrete gastrin. Enteroendocrine G cells, which they are the cells which secrete gastrin. And then release of gastrin, what does it do? It initiates the HCl release from the parietal cells and activates enzyme secretion. And it prods the parietal cells to secrete HCl by two mechanisms. So this is the gastrin. How does it cause the parietal cell to secrete HCl? Number one, by binding to the receptors on the parietal cell, and secondly, stimulating the enteroendocrine cells to release histamine. So gastrin, it directly acts on the parietal cells and causes them to release HCl, and secondarily it causes the enteroendocrine cells to release histamine. Histamine also causes increased HCl secretion. And then the third stimulation, it is the ingested protein. The buffering action of the ingested protein causes the pH to rise, which activates more gastrin secretion. So increased pH, it is increasing gastrin secretion. Increased pH mean it is going towards the alkaline. And how is this gastric phase inhibited? So the low pH, it inhibits the gastrin secretion. High pH, increased pH, it stimulated gastrin secretion, because then gastrin is going to cause the secretion of the HCl, and stomach needs that acidic environment. And then the low pH, it inhibits the gastrin secretion. When does this low pH happen? Between the meals, and it occurs during digestion as negative feedback mechanism. The more protein, the more HCl is secreted, causing decline in pH, which inhibits the gastrin secretion. So there is negative feedback in the gastrin secretion. Now we come to the next phase. We discussed the cephalic phase, then we discussed the gastric phase. Now let's come to the intestinal phase. So intestinal phase, the name is telling us that now the small intestine will be involved. So it begins with a brief stimulatory component followed by inhibition. So what is the stimulation of the intestinal phase? When the partially digested food, it enters the small intestine, it causes a brief release of intestinal enteric gastrin. Okay? But this is very brief. And this brief release, it encourages the gastric glands of the stomach to continue secretory activities. And stimulatory effect is brief and overridden by the inhibitory stimuli as the intestine fills. So for the initial phase, just as the food it enters the duodenum, there is a very small stimulation of intestinal phase, in which the intestine also secretes the gastrin. And that gastrin, it tells the stomach that you keep on secreting the gastrin and keep on doing this thing what you are doing. But when the small intestine it fills more, there is inhibition of the gastrin secretion. So inhibition of the intestinal phase, there are four main factors: distension of the duodenum due to entry of chyme, presence of acidic chyme in the duodenum, presence of fatty chyme in the duodenum, presence of hypertonic chyme in the duodenum. Inhibitory effect protects intestine from being overwhelmed by too much chyme or acidity, and inhibition is achieved in two ways: enterogastric reflex and enterogastrones. So we know the reflex, it is going to be through that. Um, let's go over. So enterogastric reflex, it is the enteric nervous system short reflexes, right? Duodenum inhibits the acid secretion in the stomach by enteric nervous system short reflexes, sympathetic nervous system and vagus nerve long reflexes, just like almost every place it is happening. And what are enterogastrones? Duodenal enteroendocrine cells, they release two important hormones that inhibit gastric secretions. These are secretin and cholecystokinin, CCK. These two, they are secreted by the duodenum, and they cause inhibition of the gastric secretion. So duodenum has two mechanisms of ending that gastric secretion. One is enterogastric reflex, other is the enterogastrons. Now this summarizes everything. Let's go over it. So this is the cephalic phase. In the cephalic phase, the sight and thought of the food and it's a stimulation of the taste and the smell receptors. It goes to the cerebral cortex in the hypothalamus and the medulla. And from there to the vagus nerve, it goes to the stomach. And now the stomach is maybe secreting something for the preparation of the food to come. And how is it inhibited? Let's also look at this. So loss of appetite, for example, if someone is depressed, the cerebral cortex, there is decreased parasympathetic activity, and then this is the negative effect, inhibition. Then the second phase is the gastric phase. So when the stomach is distended, it activates the stretch receptors. Those stretch receptors act via long reflexes via medulla and the vagus nerve. They go to the stomach, and the short reflexes. This is the neuroenteric system, the gut brain. The short reflexes, they also go to the stomach. And then the food chemicals, for example, the peptides and the caffeine and the rising pH, they also activate the chemoreceptors, and those chemoreceptors, they stimulate the G cells, and the G cells release gastrin, and gastrin goes to the in the blood, and it also stimulates the stomach to secrete its juices. Then after now the juices are being secreted in the stomach, and remember gastrin, it causes release of all uh, of the HCl, and then HCl causes the activation of the pepsin or genitopepsin, and then the movement is happening in the stomach. And now these all things, they are increasing the secretory secretions in the stomach. And now we go to the intestinal phase. Now the food is coming here, and it is starting to go where in the duodenum through the pyloric sphincter. And when it goes to the pyloric sphincter, initially in the duodenum, the gastrin is released from the duodenal cells. After that brief release of gastrin, there is inhibition. So I did not talk about inhibition of the gastric phase. So excessive acidity in the stomach, it has a negative effect on the G cells, and the gastrin release is decreased. And then when there is emotional stress through sympathetic nervous system activation, it then overrides the parasympathetic controls, and then it also, the first people, many times when they're depressed, they don't feel like eating. All right, so this is the gastric phase. Now we were talking about the intestinal phase. I said that initially, for a very small period of time, the gastrin is released by the intestinal cells, which cause the release of gastrin from the stomach as well, of juices. And then after that, there is an inhibition, and the distension of the duodenum, presence of the fatty chyme, presence of the acidic chyme, presence of the hypertonic chyme in the duodenum, it causes the enterogastric reflex, both short and long. And then the other mechanism is enterogastrons, which are the hormones secreted by the duodenum, secretin and cholecystokinin, CCK. So they have negative effect on the secretion. This is a very good table. If you go over it 2-3 times and memorize it, it will be, you don't have to go over the text again and again. So this is a table in your Pearson, and it talks about the hormones and the paracrines that act in the digestion, and not only on the stomach but on all the GIT, different GIT organs. It is good if you can go over it kind of. So just know the main effects, what are these, like cholecystokinin, and it is secreted by the duodenal mucosa, and it acts on not only on the stomach but also on liver, pancreas, gallbladder. And then the glucose dependent insulotropic peptide, GIP, it is also secreted by the duodenal mucosa in response to the fatty chymes, and then it also acts on stomach and pancreas, and it is inhibitory for the secretion of the HCl, and it stimulates the insulin release as well. Then we have gastrin. This is from the stomach. Histamine we know from the stomach. Just know the main effects, which is which of the hormones they are stimulating and which are inhibiting. And then the intestinal gastrin, it is also like the gastrin secreted by the stomach. This is this table, it continues, it talks about a few more, so you can go over it and just get like what which ones are inhibiting, which are stimulating. Let's see how hydrochloric acid, it is made by the stomach. We know that parietal cells, they make the hydrochloric acid. Parietal cells, they bring in the hydrogen ions from the blood. They absorb the hydrogen ions from the blood and they secrete bicarbonate in exchange for the hydrogen ion. And this results in increased bicarbonate in the blood leaving the stomach. This is known as alkaline tide. So for example, just to give you an example, this is a stomach, right? And this is a blood vessel which is passing by, right? What the parietal cells are doing, they are taking in the they are getting hydrogen. I'll just give you a rough schema. These are the hydrogen ions. They're getting this in because they need to make HCl, and as an exchange, they are pushing bicarbonate into the blood. So now just imagine when this blood, it is going to, from one end of the stomach, it is coming out. Just imagine this is bicarb. I'm sorry for this mess, but this is bicarb. So when this blood, it is passing through here, this becomes very alkaline. Let me write it at least so it is readable. So this blood, it becomes very alkaline when it passes through the stomach. So this is known as alkaline tide. So let me take a moment to write HCO3. So this is known as alkaline tide. And the chloride, it is pumped into the lumen, and that chloride, it joins with the hydrogen ion to form HCl. So this is bicarbonate ion. It is coming out and the hydrogen is taken in. So this blood vessel, when it passes through the stomach, it becomes very alkaline. This is known as alkaline tide. All right. Let us orient ourselves to this figure. So this is a parietal cell which is going to make the HCl. All right. So and this is the capillary which is passing by, and this is the lumen of the stomach. Now let's go from step 1 to step 4, how it is done. In step 1, we have bicarbonate ion and hydrogen ion. They're coming from the dissociation of the carbonic acid, which in turn is being formed by the carbon dioxide and water. So this is the step 1. Then carbonic acid, it is dissociated into bicarbonate ion and hydrogen. Now just keep in mind we are trying to to absorb this hydrogen ion. So this hydrogen ion, it is pumped by the hydrogen potassium ATPase into the lumen of the stomach by hydrogen potassium ATPase, because it requires energy, because inside the lumen of the stomach already there are increased hydrogen ions. So this is going against its concentration gradient. That is why we need energy. And this potassium which comes in the cell, it seeps in through the channel but back into the stomach lumen. And then in step 3, this bicarbonate, it leaves the cell, and a chloride, it comes in. So this is known as alkaline tide. So this bicarbonate, it goes into the blood, and the chloride, it diffuses through the membrane and through the channel into the lumen of the stomach. So this is how HCl is made by the parietal cell. And this bicarbonate, now this blood, when it is coming from here, just imagine if it is coming down from here, by the time it leaves the stomach, it is very alkaline. This is known as alkaline tide. Now let's go over the regulation of the gastric motility and emptying, the emptying of the stomach and the motility. How is it regulated? So the response of the stomach to filling is that it accommodates the incoming food, and two factors they cause the pressure to remain constant until 1.5 liters of food is ingested. So before this, the pressure in the stomach, it remains normal, it remains constant, it is receptive to the incoming food. So there is receptive relaxation, it is reflex mediated relaxation of the smooth muscle coordinated by swallowing center of the brain. And then there is gastric accommodation. This is the intrinsic ability of the stomach smooth muscle to exhibit stress relaxation response, and this enables the hollow organs to stretch without increasing tension and contractions. So because of these two qualities, the receptive relaxation and gastric accommodation, the stomach, it can accommodate up to 1.5 liters of food which is coming in it. And now our food is in the stomach. Now the peristaltic waves, they move towards the pylorus at the rate of 3 per minute. And where is this 3 per minute coming from? There is basic electrical rhythm. It is known as basic electrical rhythm, and it is set by the enteric pacemaker cells, which are also called interstitial cells of Gehel. Right, so there is basic electrical rhythm in the stomach, which is set by the enteric pacemaker cells, and these pacemaker cells are linked by gap junctions to the entire muscularis, and the whole of the stomach, it contracts, and in this way the food, it is mixed and it is moved, and this is located in the myenteric plexus. And this tension and gastrin, it increase the force of contraction. So these contraction, they're helping to move the food slowly towards the pylorus while mixing it. Food is moving slowly because it is being churned and mixed. These contractions, they become more vigorous and powerful near the pylorus region. So the 30 ml of chyme which is produced, it is delivered in 3 ml spurts to the duodenum. So very little of the spurt, it goes in the duodenum. Rest of the chyme, it is pushed back. So this also helps in the mixing of the food. And liquids and small particles, only they are allowed to pass through the pyloric valve and go into the duodenum. Now let's see, this is the peristaltic wave. Now just imagine this is the food in the stomach, and this is the peristaltic wave. Peristaltic wave, we know, if it starts here, then it moves to the next point, then it will move to the next point, then it will move to the next point. So rhythmically it will continue down the stomach. And this peristaltic wave moves from the fundus towards the pylorus. This is the fundus, and this is the body, and then this is the pylorus. Now the pyloric valve, it is slightly opened. So it means if this is, for example, 30, if this is 30 ml of food, only 3 ml, only a little amount, it squirts into the duodenum. Rest is pushed back. So this also causes the mixing of the food. So this grinding, the most vigorous peristalsis and mixing action occur close to the pylorus. The pyloric end of the stomach acts as a pump that delivers small amount of chyme into the duodenum. And then there is retropulsion, like I said before. The peristaltic wave closes the pyloric valve, forcing most of the contents of the pylorus backward into the stomach. So this backward movement, it results in breaking of the food and the mixing of the food. Duodenum, it can prevent the overfilling. And how does it do that? By the duodenal receptors, which respond to stretch and chemical signals, and then also the enterogastric reflex. Enterogastric reflex will be the short reflexes, and then enterogastrones. Remember the enterogastrones, the CCK, and secreted by the duodenum, that inhibits the gastric secretion and duodenal filling. Stomach, it empties in 4 hours, but in case of fatty chyme, the time can increase up to 6 hours or more. And carbohydrate rich chyme, it moves quickly through the duodenum. So that is why probably we feel more hungry when we take carbohydrate diet. So let's see how the duodenum, it controls the emptying of the stomach. So the presence of fatty hypertonic acidic chyme in the duodenum, it causes the duodenal endocrine cells to secrete androgestrons, which are secretin and cholecystokinin, and these decrease the contractile force of the stomach, and they decrease the rate of stomach emptying. And the second mechanism, it is chemoreceptors and the stretch receptors. They trigger the enterogastric reflex, and they use both short reflexes and the long reflexes. Short reflexes, they act via the gut brain, which is the enteric neurons, and the long reflexes in the CNS, there is increased sympathetic activity and decreased parasympathetic activity, which causes the decreased contractile force.